Okay, so welcome back to this next video in which we are discussing angiogenesis in chronic inflammation. Now we're just talking about the building up of the immune response. So we've talked about the acute inflammatory response, and we're now talking about the adaptive immune response. So we've discussed that pathogens will have these antigens, okay? And an antigen is just a name for anything which is capable of having an antibody made against it, okay? And these antigen-presenting cells will take antigens, and they'll do this for a huge number of different antigens, but we're just considering a single antigen now. And they will take this antigen, break it down into fragments, okay? And they'll present these fragments on their surface. They'll then go to the lymph node and use these fragments to activate CD4 positive naive T cells. And at the moment we're considering a humoral immune response, so a response to an extracellular pathogen, okay? And then what will happen is these CD4 positive naive T cells will then become activated and will differentiate into T helper naught cells. Okay, the T helper naught cells will then proliferate, okay, and you'll end up with an entire population of this T helper naught cell, and all of these T helper naught cells are specific to this initial antigen fragment, so they are all targeted against that specific antigen fragment. Then they will start differentiating into T helper 2 cells. This is specifically in the humoral immune response. Now, concurrently with this, you're going to have B cells becoming activated. So within the lymph node, you have a huge number of B cells as well. Now, these B cells are all specific to a certain antigen, okay? So every single B cell is different, and it will produce antibodies that are targeted against a specific antigen, basically. Okay, so when this antigen that we are discussing now, remember we chose a specific antigen, when this antigen, and in fact I'm going to draw this antigen, so let's say here is this specific antigen, which I'll draw as a blue blob, which we want to launch an immune response against. Okay, so when this antigen this antigen will be released into the interstitial fluid, either by the pathogen itself or by neutrophils which are phagocytosed and destroy the pathogen. Then what will happen is that these antigens will go through the lymph vessels to the lymph nodes, and then they'll find the B cell. If they do manage to find the B cell which has a receptor on its surface which is perfect for this antigen, then what will happen is that B cell will then be activated. However, it won't be activated just yet. Instead, it needs to interact with the T helper 2 cell. Okay, and if the T helper 2 cell and the B cell interact correctly, and remember this T helper 2 cell is also specific for this antigen here. Okay, so if the two of them work well together, then what will happen is this B cell will start to produce a huge number of plasma cells. Okay, so here we have plasma cells. And these plasma cells will produce antibody, basically. Okay, so let's produce a whole population of these, and I'll just label them as P from now on. Okay, so here is our population of plasma cells. Okay, so these plasma cells will then go into the blood and start chucking out antibody which will bind to the antigen, and if potentially that antigen is a toxin, the antibody will neutralize the toxin. So these plasma cells are going to produce antibody, so let's draw antibody here. Okay, so antibody has this sort of structure where it has 2 heavy chains. Okay, so I'll label this up as antibody. So they have 2 heavy chains, which I'll show here in orange. Okay, one here and one here. And those 2 heavy chains are linked by a disulfide bond here. And then also 2 light chains. And these light chains are attached to the heavy chains, again by disulfide bonds. Okay, so I'll show the light chains in turquoise here. Right, so plasma cells will start producing antibody, basically. And antibody has three main functions. Firstly, if the antibody is against an antigen which is actually a secreted toxin from the pathogen, then the binding of the antibody to the antigen will neutralize the toxin. Okay? And I should have said that actually. Antibody's principal function is that it now binds to this antigen which the B cell was specifically targeted against, and also this CD4 positive T cell was armed against. Okay, so it's going to cause neutralization of toxins. Alternatively, the antigen that we've produced this antibody against, it could be on the surface of the pathogen, and in that case what will happen is the antibody will attach to the surface of the pathogen. Now, basically that has two functions. Firstly, if you've got antibody on the surface of a pathogen, that can activate the classical complement cascade. So there is a complement pathway which makes use of antibodies on the surface of pathogens, and basically that's the triggering structure which causes this classical complement cascade. Okay, and the classical complement cascade will then lead to the destruction of the pathogen. Okay, and finally, you also get opsonization, and this is a fantastic word. So, when the antibody binds to this antigen on the surface of the pathogen, then basically phagocytes such as neutrophils and macrophages have receptors for this fixed portion down here, and I should say that this portion up here is the portion which binds to antigen, and this portion down here is a fixed portion, so the antigen will bind here. Okay, so basically if the antigen is on the surface of a cell, let's say here is the cell membrane of the pathogen, then the antibody will bind onto the antigen, and then it will have this dangling bit down here known as the fixed region, or the FC region. Okay, and phagocytes such as neutrophils or macrophages have receptors for this fixed region of the antibody, and they will bind to those, the receptor will bind to this fixed region, and basically that then triggers the phagocytosis pathway. So remember when I was telling you about phagocytosis, I said that initially the microbe needs to bind to the phagocyte. Well, this is one of those interactions whereby the phagocyte can bind to the microbe, basically. Okay, so having these antibodies attached to the surface of the microbe will increase the probability that it's going to be phagocytosed, and this increase in the probability that it will be phagocytosed is known as opsonization. So the antibody-antigen complex on the surface of the microbe is going to increase the chance that the microbe will be phagocytosed. Okay, so that is the humoral immune response. That is how we attack extracellular pathogens. In the next video, what we'll do is turn our attention to how we're going to attack intracellular pathogens through the cell-mediated immune response.